Welcome to Conversations. I'm Scott Turner, and my guest today is Professor Addy of the Department of Chemistry at Ben Gurion University of the Negev. Addy is a chemist who's interested in the interface between biology and chemistry, especially with respect to the problem of the origin of life. Addy, welcome to ESF.
Thank you, Scott. Good to be here.
I'm glad you're here. Now, chemistry and biology. Now, we've been in the midst of a chemical revolution in biology for about the past 60 years now, probably longer, and some biologists regard that as a great positive, because we've learned a great deal about the way life works at a chemical level. Other biologists are a little bit concerned that biology is disappearing as a distinctive science, that it's becoming a subdiscipline of chemistry. And you're right there at the interface between the two. So I wonder if you could tell us your thoughts from a chemist's perspective.
Yeah. Well, it's actually very interesting. A paradoxical situation seems to have arisen in that despite the enormous advances in molecular biology, where we seem to know more and more about how the cell works in chemical terms, every couple of years, another Nobel Prize for that sort of work. We've reached a strange situation where we know more and more about the detailed machinery, but we're still struggling to somehow connect between chemistry and biology. And maybe the most dramatic way to show the problem is to express what a great biologist, Carl Woese, said not too long ago, that biology at the beginning of the 21st century is where physics was at the beginning of the 20th century, before Einstein, Schrödinger, Bohr revolutionized the subject. So it's a very strange situation where on the one hand, because all of the developments in genomic understanding, for example, we have a feeling that we're almost there. And on the other hand, a great biologist who's worked in this area comes and says where before a revolution, the subject has to totally undergo some change in order to be properly understood. So there's a paradox there.
That's a pretty provocative statement. So where do you think the revolution is going to come? I mean, what are the points of conflict, if you will, that are going to drive this revolution forward?
The problem is that there's a growing appreciation of the fact that biology is not just an array of things, living things, but it's a process. A continuing process, and that process actually began with, let's call it the origin of life or the emergence of life on Earth, and that means that somehow chemistry and biology have to connect, and connect not in just a theoretical way, but connect in the sense that it was an actual process, that at some point we had chemistry, and that chemistry evolved in a certain way and became biology. Now, what's interesting here is in the last, let's say, couple of decades, a new area of chemistry has started to develop called systems chemistry, and that area is beginning to fill the void between, let's call it regular chemistry and biology. Systems chemistry deals with replicating molecules and the networks that they establish. And that is filling that gap between regular chemistry, which generally doesn't deal with replicative systems, and biology, which can be thought about as a complex kind of replicative chemistry. So if you want to connect chemistry and biology, you have to fill that void with simple replicative systems and how they work, and lots of very exciting work is now being done in that area, and a picture is beginning to emerge that will enable us to understand the process by which chemistry became biology.
So, the notion that the cell is a very complicated chemical system is obviously the case from, the more we know, the more complex it becomes. But how does that make biology stand out from chemistry? Complex chemistry is still chemistry after all, and the concern, of course, is that amongst biologists is that we are going to all become chemists in the end. That the distinctive nature of life that seems obvious to the senses is going to be swallowed up by chemistry, basically. So what's special about systems biology that is alive?
Yeah, I think the key here is that because biology is so complex, it's very difficult to seek out its essence, because the essence is, if you like, is buried under layers of complexity. And the analogy I like to use is that of two airplanes, the Wright brothers 1903 model and a Boeing 747. If you want to understand the principles of flight, let's say you don't know anything about flight and how that operates, you don't want to try and figure out the principle of flight by looking at a Boeing 747 with its kilometers of wiring, millions of parts, TV screens, ovens for heating food, etc. It's very, very confusing. Where do you start? But go to the Wright brothers 1903 model, and everything there is essential for flight, and you can understand the principles more readily there. If you want to understand how something works, look at a simple model first. So systems chemistry is saying, if we can look at the way that simple replicating systems operate, behave, what are the rules that govern their way of behaving and their evolution, that's very important, we may understand many fundamental biological issues that at present are hard to access, to comprehend, because they're buried in complexity.
So the analogy to the Wright brothers, to the Boeing, I guess the latest one is 797. It underscores a very controversial question in evolutionary biology, namely this evolution of complexity, and as a bone of contention amongst many biologists, whether life evolves generally from a simple to a complex state, and what drove the evolution of the airplane. Obviously, there are engineers and other kinds of people involved in pushing that design towards a more complex state. Yet, what is it about evolution that necessarily would drive it towards a more complex state? What do you think makes that distinctive?
Okay, this is a fundamental question, and it comes down to this, that the original idea that you have some chemical system that somehow became simple life in a chemical phase, and then once life emerged, simple life, it then began evolution in the Darwinian sense. That idea is increasingly looking wrong. Inadequate. It's not two separate processes, a chemical process followed by a biological one. It's one continuous process, and one has to redefine the terms of reference in order to incorporate or to show that we're talking about one continuous process. And in order to do that, one has to talk about the whole process in non-biological terms, so that it encompasses chemical systems as well as biological ones. And a central idea to do that is to talk about the concept of dynamic kinetic stability. Now that sounds like a mouthful, but it's basically saying that there is another stability kind in nature that may have been overlooked, and that's the stability of things that can make more of themselves, and this has to be defined in physical or chemical terms rather than biological terms.
Okay, that raises a very interesting point about how life becomes life from chemistry. We'll be back in a moment with more conversations with Addy. Welcome back to Conversations. Our guest today is the chemist/biologist Addy. So Addy, in the and when we left off, you were talking about what seems to be a very important concept, namely dynamic kinetic stability. And this enters into a debate that's roiled origin of life research for decades. And if you look at living systems, you see two things that are distinctive about them. One is that they have metabolism, that is, they organize matter and energy to create themselves, and the other is that they replicate, they make copies of themselves. And this has split origin of life research into two schools: metabolism first and replication first. And no one has ever actually been able to get to the finish line, namely, what's the origin of life? And you're saying in some of your papers that you've resolved that issue.
Well, I don't want to say that I've resolved it. I believe it has resolved.
Yeah.
And this is coming about through our deeper understanding of the process and through the recent developments in systems chemistry. The debate originally was, as you put it, what came first? The replicative ability, a genetic molecule, for example, or complexity? Complexity as manifest through metabolic capability. But already, I would say, for a number of decades, the metabolic first school has changed direction a little in incorporating an autocatalytic.
What do you mean by autocatalytic?
Okay, autocatalytic meaning the ability of the system to catalyze the formation of more of itself. In other words, autocatalysis is the fancy chemical word for replication. Mm-hmm. They're not entirely identical, but for our purposes, close enough. So an earlier idea was that through a complex kind of system, you could generate, through a cycle, a system that made more of itself, as opposed to the genomic, the replication first approach, which saw the essence of replication as being in a molecule and the sequence, like a nucleic acid, and its sequence. But what's become clear is that both of those approaches depend on replication or autocatalysis, if you want to use the other word. So in that sense, they are to a degree overlapping. But what has become beautiful is that recent work in systems chemistry is showing actually that both schools were right in a way. And the reason is that a single replicating molecule is very ineffective in making copies of itself. But once you make a little cycle of these genomic type, if you like, sequence molecules that have a specific sequence, nucleic acids, DNA, RNA, that sort of molecule, then the process of replication is facilitated through having a cycle. So a small network of these RNAs, for example, will be more effective at making copies of itself than a single molecule, and therefore both schools can claim we were right all along, that the two have merged into one unified view of the problem.
Does a self-replicating molecule have to be something as complicated as a nucleic acid, though? Can it be something else?
Well, it's not just nucleic acids that can make copies of themselves. Some peptides are also showing this capability, and there are some other synthetic molecules that can also do this. So actually self-replication is, I wouldn't say it's a common characteristic amongst chemical entities, but it's not restricted to one family.
So it can actually go beyond this question that's bedeviled the origin of life for a long time, namely that the origin of the gene, or to use a broader term, some form of hereditary memory, is not limited to a complex molecule like RNA or DNA. It can be any kind of molecular system that has the capability of mobilizing energy to be able to reproduce itself, to make more copies of itself. Is that a fair way of putting it?
Yeah, I think so. I would say the following. We're not in a position to look back billions of years and say what actually happened. These are historic questions. And the origin of life debate or research is now moving, I think, in a more productive direction towards understanding the principles by which inanimate matter would want to complexify towards life, rather than what happened where a long time ago, because basically we will probably never know those historic questions, just like we don't know what Napoleon had for breakfast, on his 18th birthday, and we may never know.
Yeah, but you're making really a very radical statement that's going to make a lot of biologists uncomfortable, and that if you go back to these kinds of self-replicative autocatalytic systems as the foundation for the origin of life, you're really proposing that there's this inevitable trend of these systems towards greater and greater complexity. And many biologists would actually take you to task on that issue.
Yeah. Well, I think, as I say, through this area of systems chemistry, we can understand the connection between three central elements: function. In biology, we see function wherever we look, stability, and complexity. Now, it's very clear if we look around us in the world that function is improved through complexity. That Boeing 747 that I spoke about is much more complex than the Wright brothers plane, and that complexity wasn't put there for no reason. It was put there, it was incorporated there in order to improve function. Well, guess what? Nature has understood that very same principle, and replicative function of a molecule or a small network of molecules is very fragile and very ineffectual. So nature does exactly what the engineer in building his planes and evolving those planes does. Nature has complexified that simple replicating system in order to improve function, in order to increase replicative stability. Because ultimately the name of the game is a persistent replicator, stable in the sense of being persistent, not stable in the thermodynamic sense. And therefore this stability is induced, this persistent stability is induced through complexification. So the process was an inevitable one of complexification from some fragile replicating system that will only replicate in the lab when you've got a chemist and postdocs and fancy equipment that costs millions of dollars or hundreds of thousands of dollars. Whereas the bacterial cell doesn't need any chemist or biologist or postdocs or whatever. It happily replicates away in your backyard, in just about anywhere else. Now that robustness, that versatility has come about through complexification, just like in the Boeing 747.
Okay, you're raising a very radical question, namely the chemical origins of core Darwinian concepts like fitness and selection, and we'll come back to these questions right after the break with Addy Pros. Welcome back to Conversations. Our guest today is Addy Pros from Ben Gurion University of the Negev in Israel. So, Addy, in the last segment, we were getting into some really kind of radical statements about complexification and the inevitability of this, and we're really getting into really redefining Darwinism in chemical terms. For example, the old idea of fitness, it's very tied into this replication idea to the exclusion of metabolism. But what's the implications are of what you are saying is that they are actually merged, and we have to think differently about things like fitness, what natural selection is, what's being selected, those sorts of things. So and you have some interesting thoughts about how those two things merge, especially at the origin of life.
Yeah. Well, the Darwinian terms, fitness, natural selection, and so on, are biological terms, and they only apply to biological systems. So in a sense, they're not adequate for defining or governing the entire process of a chemical system that became a biological one. And we need to basically utilize more general terms and the more fundamental terms, which are chemical terms. So instead of natural selection, in chemistry we talk about kinetic selection, which is basically a fancy way of saying the better replicator will tend to win. But I think the most fundamental change is this very elusive and problematic concept of fitness can be replaced by the term stability, but not the stability that we're familiar with normally, thermodynamic stability, but this dynamic kinetic stability, the stability of things that are able to make more of themselves and are present, abound, because of their ability to make more of themselves.
So
persistence.
Just to clarify things, when you're talking about thermodynamic stability, you're really talking about an equilibrium state. That is one that the systems tend naturally towards, and they stay there unless you put energy in. But you're really talking about a very different kind of stability, which is inherently a disequilibrium, and that seems to me to be the core concept that you're trying to get.
Absolutely.
Right?
This is not the thermodynamic stability, the equilibrium stability. This is a non-equilibrium state, a far from equilibrium state, whose stability is manifest through its persistence. Cyanobacteria are very persistent in one, with very little morphological change. They've been around maybe for, you're the biologist, you'll know better than me, whether it's 2 billion years or 2.5 billion years, but for a long, long time. Now that's persistence. Mount Everest has been around for 60 million years. That's small change compared to the cyanobacteria. So a replicating system seeks to become more persistent because in the world of replicators, and that's what we're learning through systems chemistry, poorer replicators tend to be transformed into better replicators through complexification. So this is the process that we can understand evolution, Darwinian evolution, in non-biological terms. An unstable, in this dynamic kinetic sense, in this persistent sense, an unstable system increasing its complexity to become more and more stable through complexification. And incidentally, complexification isn't just something that manifests at the individual level, the complexity of a prokaryotic cell or a human being or a dog or whatever. It's the complexity manifests itself through network formations. So any so-called individual living entity is not really fully individual. It is intricately connected through a network with other living entities, not necessarily its own kind. As you well know, 90% of the cells in our body are actually bacterial cells. So every human being is, in a sense, an ecological network.
Yeah.
So individuality is actually starting to get lost through this process of complexification.
The interesting thing about that, especially to a biologist here like myself, is that you're really talking about fitness as persistence of these dynamically kinetically stable systems. That's a radical redefinition of our concept of fitness. But the most radical thing that you're saying is that fitness scales across all levels of organization of life that we can imagine, from the replication of complex molecules within cells or at the origin to the entire globe, really. And
well, there's a lot to what you say there, and in fact, there's a growing, I mean, we can't help having to agree that there is a process, an ongoing process by which non-replicative matter, regular chemical stuff, if you want to call it that, is being converted into replicative stuff. Initially, with the origin of life, we believe all the matter on Earth was non-replicative, and in some location, don't ask me where, I have no idea, and under conditions that I cannot specify, we don't know, a fragile replicating, persistent replicator emerged and then proceeded to evolve, to complexify, and to extend itself basically over the entire planet and transform non-replicative matter into replicative matter. The implications of that are really quite staggering, and one really wonders where that's actually taking us. But that's already a big question that I don't want to answer.
Staggering and big is certainly the word for it. I wish we had much more time to explore these issues with you, Addy, but we have to stop now. And thank you for coming to ESF. Thank you for joining us in Conversations. And thank you for joining us in Conversations. This is Scott Turner saying goodbye, and we're glad you joined us. We'll see you next time.